I'd like to welcome everybody to the 25th Norman Geschwind Memorial Lecture. This is a series of lectures that commemorates Norman Geschwind, who was the first chair of the neurology department at Beth Israel Hospital. And Geschwind was the James Jackson Putnam Professor at Harvard and started at City Hospital, where the neurology department was located at the time. And when Harvard moved from City Hospital in 1975, he brought his department over to Beth Israel, and then he proceeded to make it into one of the great places in the world for behavioral neurology until his untimely death in 1984. And the department put together a fund to allow us to have an annual Geschwind visiting professor, and we've been doing that since 1994. And you can see the previous speakers on the back of the page. You'll note that the last lecture was given in 2017, not 2018. And the reason for that is that we invited Dr. Buckner last year to give the lecture. And just before the lecture, he had a orthopedic incident, which made it impossible for him to give the lecture. And so we decided that we would just cancel it for that year and invite him back this year. So this is the 2018 and 2019 Geschwind lecture. It is a special pleasure, though, to be able to introduce Dr. Buckner here today. He received his training at Washington University in St. Louis through his PhD degree, and then was a Charles A. Dana Foundation postdoctoral fellow there, where he worked with Dr. Mark Rakel. And I believe that Mark was also one of the previous speakers in this series a number of years ago. That laboratory produced a continuing stream of leaders in the field of cognitive neuroscience, of which Randy is one. He then became an assistant associate professor at Washington University and was collaborating with investigators at MGH who realized how indispensable he was and enticed him to move to Harvard University in 2005 as a professor of psychology and neuroscience. And he also became a Howard Hughes investigator at that point. And he is the director of the Psychiatric Neuroimaging Research Program at MGH since 2008. Dr. Buckner is best known for his work using functional connectivity to identify networks in the brain for specific significance in terms of behavior. His work has permitted us to see an overview of the functional organization of the human cerebral cortex and to relate that to the gyral anatomy of individual people's brains. And it's helped us understand how this relates to a variety of neurologic diseases, particularly Alzheimer's disease, and how these diseases affect the cerebral cortex. And it's given us insights into memory, executive function, and the concept of the self. This body of work consists of over 300 publications now. It's been cited over 60,000 times. That's a lot. With an H-index of 110. That's also a lot. And he has received a series of awards from the time early in his career when he received Young Investigator Awards from the Human Brain Mapping Society and from the Cognitive Neuroscience Society, and then later the Troland Research Award from the National Academy of Science and the MetLife Award for Medical Research. He's a fellow of the American Psychological Association and the American Association for the Advancement of Science, and has served on the editorial boards of a range of journals, including Neuroimage, Nature Reviews Neuroscience, Cortex, Neuron, Journal of Neuroscience, and others in this field. He has been an invited lecturer at numerous national and international meetings and has held many named lectureships, and we welcome him as a 25th Geschwind visiting professor.
Well, thank you for that warm introduction. It's a true pleasure to be here today because it's not often that one gets to give a lecture that honors somebody who is so directly inspirational to the work that's going to be talked about. And so this is interesting. When I put together the talk today, I wanted an introduction that was appropriate to the work of Norm Geschwind. This introductory slide is one I've used in other lectures because it is a good introductory slide to the work. Norm Geschwind, I think, is been such a significant figure because specifically of the things he taught us and how it carries on and sets the foundation for all of the things we've been doing, and also the basis of modern systems neuroscience. What he taught us is how to link anatomical understanding and circuitry that you can see in animal models to syndromes that you see in humans. And in doing so, he taught us a basic tenet of systems neuroscience, and that is multiple regions of the brain work together because of how they're connected in the service of mental function. And critically, and this is where I think I'm going to take off from in my own talk, circuitry that underpins faculties that have evolved and have special role in human cognition, such as language, including his seminal papers where he talked to the wide-ranging audiences about how circuitry of language could be understood in terms of the neurological syndromes, but also just in terms of helping us to understand how the brain does such an extraordinary faculty. And so in that spirit, I'm going to jump off and actually speculate a bit about some of this circuitry, even how it arises and what we've learned from his foundation. So it's really humbling to actually come, and I wish you were here in the audience to give me feedback, to tell us if you thought we were on the right track or all the places we've gone awry. So what I want to do is just draw your attention to a contrast, because I think this is a critical contrast that has been underemphasized and provides insights into how we think about higher level cognition. And the contrast is an anatomical one. So to the left over here is a early diagram. There's now much more sophisticated, more complicated ones, but the point is captured in this critical early diagram from John Mansell and David Van Essen, showing the anatomic connectivity from the primary visual cortex to secondary visual cortex in extrastriate areas such as MT, which is a motor and selective visual extrastriate region, all the way to the parietal areas, and then to the frontal cortex, the frontal eye field that drives motor behaviors. This is a canonical sensory motor hierarchy by which sensory information from the outside world is taken in, transformed, and processed in a cascade that eventually can be transformed and guide motor movements such as that you see in the frontal eye fields. And this is the model that most of systems neuroscience uses to understand how animals and humans respond to the here and now, how we take information from our outside world in a context-relevant way, processes it, and use it to drive motor behaviors that help us survive the moment. And it's in some sense intuitive. Here you have sensory information coming from the retina, the sensory epithelium. It's driving a cascade patterns and constraints. We've learned a lot about how the brain has top-down processes and context that shapes that incoming information so that we can use it. And we can see the immediate relevance of how it can guide a motor action in this moment that relates to that input. What I would like to argue in this talk is that most of the primate brain, and in particular in the human brain, is not about circuits that survive the here and now. That is not about circuits that take in their main constraints from the sensory epithelium to drive a motor action. And further, that the circuits of thought, well, I'll show you how they have family resemblances to these more ancient circuits, and you can see how they could have evolved from them. It's not that they're in between, advanced in between processors from the sensory world to the motor output, but rather parallel circuits going on largely on their own, independently, and have the properties that allow us to drive thought. And there's actually precedent for this anatomically in a beautiful paper I encourage all the students to read from Pat Goldner and Keish, who noted this. And she was enabled with another anatomical breakthrough at the time, where she was enabled by not single tracer techniques, but double labeling techniques, where you could look at two regions of the brain, make an injection in the macaque, and actually simultaneously see all the regions that project them. And what she noted when she looked at association cortex, prefrontal area 46, and this parietal association area 7A, that when she made injections in these two areas, which she knew to be connected, it wasn't the case that they looked as if there are two levels in a hierarchy by which the higher area was projecting further into the system, than the earlier area. What she noted is that they actually projected to upwards of 15 regions that were in common. And the projection patterns weren't of the classical hierarchical kind you see, or didn't have the same rigid hierarchy, is probably the more specific description, as if all of these regions were talking to each other in a reentrant circuit and without any other information. That's an interesting anatomical property. You have regions across the brains projecting to each other without a rigid hierarchy. If one imagined what a circuit of thought might look like, that you'd have activity pinging about, potentially even detached from the sensory environment or motor actions, this would be that circuit. And what I'm going to argue is I think she got it right largely, and talk about our modern understanding of these circuits, and to some degree, how they're similar and different from these canonical sensory motor hierarchies. And so just to summarize the talk, I'm going to dive into data suggesting that there exists a motif for cortical circuits of higher cognition. I'm going to provide some evidence for that anatomical basis in the marmoset monkey. I'm going to show that motif is expanded in humans into multiple juxtaposed circuits specialized for language and social functions. I think it's really interesting to think about how these, extraordinary abilities in humans could evolve their extraordinary capacities, but do so under the constraints and from the same basic circuit properties that have existed for many tens of millions of years. And then I'm going to speculate on how developmental processes might form those circuit specializations. And so to do this, I have to step back further in time. Here is a plot of human brain evolution over the last several million years. And there's an interesting facet that is, I think, probably the most defining feature of our particular niche in evolution. And this is a plot of brain size from fossil endocasts going back 6 million years. And like most other primates, there's a general trend towards larger brains holding all else equal. You can see that was going along for a period of time to about 3 or 4 million years ago. And then you see something that defines the human niche. Our brain size tripled over about 2.5 million years. And you could have a whole lecture, many lectures, on what it might have been the gateways to allow that, the changes in their diet, the pressures in terms of social environment. But what happened over an extraordinary short period of time is that the brain, and the cerebral cortex in particular, expanded vastly, tripling in size over an evolutionary short period of time. And what happened during that evolution? And we don't know. We don't have a record of the soft tissue, but you can make some estimates from homologies from living primates. And here is a picture of the architectonics looking under a microscope of the cell distributions from Corbinian Brodmann's original work, where you can see a monkey. They're not drawn to scale. The human brain is about 25 times the size of this monkey brain. And you can see areas. And the first thing you'll notice is that many of the areas under a microscope look very similar. Here is the primary visual cortex in the back of the head. Here it is in the back of the head of a human. The other thing you'll notice is that the primary sensory cortices and the motor cortices make a relatively smaller portion, define a relatively smaller portion of the human brain compared to the monkey. And over here, area 17 takes up a good portion of the brain. Here, it's a very small region. There are secondary areas, such as area 7 here, which might be a sensory motor transformation zone by some accounts, and you see it over here, those homologies. But then you get a number of regions in association cortex, area 39, area 40 here in parietal association cortex, multiple prefrontal areas, and regions in temporal association cortex that Brodmann himself was uncertain or didn't think had homologies in the monkey. I think there are strong homologies to all of these areas and circuits in monkeys, but they're either new by Brodmann's account or by what I believe vastly expanded. The other thing I want to do is just to spell for the students. We often talk about expansion and evolution and talk about prefrontal cortex being disproportionately expanded. Well, prefrontal cortex is disproportionately expanded in apes and in humans. Probably the better description is that association cortex all around the brain is disproportionately expanded, including parietal association cortex and temporal association cortex. All of these regions between the sensory motor zones come to take up most of the territory in the larger primates and much of the brain in the human, which is interesting in and of itself. It suggests a very simple mechanism that happens when the brain expands, not a extraordinary evolutionary event that expands this, and that. Rather, it probably just happens when you scale up of the brain. And so one of the questions we have is, what does the circuitry of these association zones look like in the human? And the insight we had was by homology to monkeys in the past, and also the extraordinary insights of dissecting that circuitry from the lens of neurological syndromes, as Geschwin exemplified. Recently, there have been newer methods, and you've heard quite a bit about these because they're well represented in your department, for measuring brain networks in the human. The first thing I would say for the students is that they're very crude. If you asked us what kind of method we would like to have, how precise we'd like it to be, we would want what all system neuroscientists and molecular neuroscientists want: extraordinary cellular precision to be able to see all the connections and the full connectome across the brain at will in humans. But we don't have that. What we have is incredibly crude macro scale methods that are indirect proxies for circuitry. So one thing that's been surprising to me over the last 15 years is just how far they've taken us to unravel organization in the human brain, given how indirect they are. And there's two main methods. One of them is based on diffusion MRI imaging, shown over here. I'm not going to talk about that technique, that allows us to see the large bundles, the fascicles across the brain using water diffusion properties exploited with advanced MRI sequences. The method I'm going to be talking about is intrinsic activity. And it has to do with the fact that there's a tremendous amount of spontaneous activity in the brain. This exists at multiple scales. If you look at individual neurons, you'll see spontaneous activity at columns and at the level of large-scale brain circuits. It's interesting to think about what the origins of that spontaneous activity are. They're almost certainly subserving very low-level functions. They exist in all states: task states, wake states, passive states. They exist in sometimes changing forms under anesthesia, but they are pervasive. They also exist across all of the systems simultaneously in the brain. They're not special to cognition. They're not special to any circuit in particular. This is a broad general property of activity cascades in the brain, tremendous spontaneous activity. That they're there creates an experimental opportunity to use them to measure organization. And that's because anatomically connected regions, on average, tend to correlate together in this spontaneous activity. The original demonstration from Murat Biswal's laboratory, he took us what I'm referring to as a seed region here, and he measured spontaneous activity in the hand motor region. And here's an example of that activity over many minutes. And he simply asked across the brain, what is correlated to that hand motor region? And what he saw was the well-known distributed motor network. And this was just an existence proof, a proof of concept that spontaneous correlations across the brain could identify known circuits. This was then taken quite a bit further by people like Michael Grecius and Michael Fox, who is now in your department. This is when he was a graduate student, sort of launched the field forward by showing just how powerful these methods were to unravel many systems in the brain. And so I'm going to pick up on that foundation and show you how we've been able to use it. But before I do, I also want to show you a couple of validations. These are indirect methods, I said, and there was also a lot of question about what they might reflect in the kind of confounds. There is vasomotion vascular effects that could be at the origin, and these spontaneous correlations might not actually be specific to anatomy, but could rather sort of anatomic connectivity, but could also be reflecting, for example, vascular distributions and properties. And so it was critical to validate that these methods could tell us details, and as we expect, in systems where the anatomy is well known. And so we conducted a long line of work, actually looking at cerebral cerebellar circuitry. It specifically started because we wanted a system to validate these methods, but it turned into its own research program, which I'm not going to have time to talk about today. But cerebral cerebellar circuits are interesting because they have very precise anatomy worked out by people like Jeremy Schmaman, by which the cortex projects to the pons and then projects predominantly contralaterally to the cerebral hemispheres. The feedback projections go from the deep cerebellar nuclei up to the thalamus and back to the cortex. And as Peter Strick's work has shown, in a closed loop circuit, such that the regions of cortex that project to the cerebellum receive projections back. This is a beautiful circuit for validation. It has very idiosyncratic anatomic projections to specific zones that go as far across the brain as you can go and across vascular distributions. When you take a seed region in the hand motor region and look at its spontaneous correlations, you can beautifully see the primary motor representation, and for those who are cerebellar connoisseurs, the secondary motor representation that's been known for a long time. And here's the contralateral. So this in of itself, we felt suggested that we're on the right track, that these methods could see large-scale circuitry in the brain. Another validation came from scouring patient records. This is something that Sheng Liu did with Lucy Liu, finding very small hemipons lesions, which the anatomy has been worked out. Hemipons lesions disrupt the contralateral projection zones but leave the ipsilateral projections intact. This is a beautiful kind of system to work with because it makes a counterintuitive prediction. It predicts that regions near to the stroke will show preserved correlations, but regions away from the stroke will show impaired if, in fact, anatomic circuitry is the driving force behind these correlations. That's what you find. If you look at left hemipon lesions, you disrupt the contralateral connectivity but leave intact. If you look at a group of individuals with the reverse lesions, you'll see the opposite. Again, suggesting that these methods are picking up and allowing observation of anatomic circuitry. You can also use cerebellar circuitry to look at specificity. At this time, Lord Adrian, after he'd won his Nobel Prize for the basic physiological work that he did, he actually, for those of you who know the history of cerebellar anatomy, was the first to work out the accurate representation, the somatomotor representation in the cerebellum, and that's because there's no direct anatomic connectivity from the cortex to the cerebellum, so degeneration approaches and techniques, even retrograde labeling and anterograde labeling techniques don't work for cerebellar circuitry. It was actually worked out taking advantage of physiology, recording along the motor strip and stimulating the motor strip in the hand, foot, and mouth regions, and then looking and seeing where you got evoked response in the cerebellum. Adrian was able to work out this inverted topography that you see on sort of the dorsal surface of the anterior cerebellar lobe. You can do that experiment in humans. Here is a person in the scanner. Here we're asking them to move their foot, we're asking them to move their hand, and here we're asking them to move their tongue, and you can make a beautiful motor map. You could see that inverted representation of cerebellum. If you have 1,000 subjects lie in the scanner and record spontaneous activity, this is a difficult study because the signals are weak, so you see it very cleanly with large numbers of subjects, and you look at this spontaneous activity in the foot region, the hand region, the mouth region, you can make that same map just from the spontaneous activity. Again, suggesting the anatomic connectivity is correlated with the spontaneous measure we're able to make. And so I just wanted to show these validations because there are often questions about these techniques and what they reflect. At least is analyzed in this way at this gross macroscopic level, they're able to reflect details of known anatomy even across widespread regions, across vascular distributions. And so we can take these techniques and now use them to start to unravel cortical zones where the anatomy is unknown, especially those zones of a higher order cognition and association cortex. And so what I want to do is show you the results of a first experiment where we looked at canonical sensory motor hierarchies. And so what you're seeing here is correlations with spontaneous activity in parietal association cortex across the brain. This is the human homologue to the macaque area LIP, we think. And what you see is regions in extrastriate visual cortex and multiple zones that we think are the human homologues of the frontal eye fields. This is a not surprising pattern. If you look at the correlation strengths across these areas, you actually get a network that is very similar going from primary visual cortex up through parietal association cortex to the frontal eye fields that recapitulates what's been long known from macaque anatomy. Again, this is a form of validation, but here in cortex, showing that we could recapitulate the known organization of the canonical sensory hierarchy. Where things get interesting is if now you take the experiment from the known circuitry into these unknown territories of association cortex. And so just as a reference, I'm going to take this network shown here, which is the canonical sensory motor hierarchy, and I'm going to project it onto the brain as a reference over here to the right. In the human literature, this is something that Mike Fox has studied extensively. It's often known as a dorsal attention system. In the macaque literature, it's often referred to in variations of the oculomotor system, and it's been well studied in macaque physiology. The experiment that I'm going to do is actually to then move this seed region all the way up and through these regions of association cortex. And I'm going to do it as a movie so you can see the organization change, but it's actually not a dynamic movie. It's an experiment done dozens and dozens of times. I'm just making into a movie so that you can see it. And I want you to watch a couple things as that moves. The first thing I want you to notice is that as that seed region moves into association cortex, you can see these rather dramatic shifts in the circuitry. And so that, a region of association cortex clearly have multiple association regions going through each other to nearby zones. The other thing you may have noticed, and you can see it here when you look on the left compared to what you're seeing on the right, is that while there's shifts in topography in the anatomic organization, the basic motif is the same. Each of the parietal regions that you go through tends to have a frontal projection zones, tends to have a temporal projection zones, and if I showed you it on the midline of the brain, you'd also see this similarity. In fact, if you look at detail, and we've done this both from marmoset anatomy and in humans, there's actually a gradient of networks that go rostral to caudal. Caudal to rostral, the frontal cortex, caudal to rostral and temporal cortex, and there's a gradient in parietal association cortex. This is interesting from an evolutionary standpoint. It looks like Mother Nature didn't invent new circuitry. As the brain expanded, it duplicated the same canonical motif over and over again. But the third point I think is the most relevant for this talk. While this canonical motif is the same, when you go into this zone of parietal association cortex, which is probably in the macaque what people call OPT, it is not coupled to sensory or motor regions. It itself is only coupled to distributed association regions that are as far on the cortex as you can be from sensory and motor hierarchies. I think this is the great, development in primate and human evolution. Maybe somewhere in the smaller brain regime, we solved the here and now with sensory motor circuits, and as the brain expanded, there's opportunities for those circuits to expand, and they got to explore a completely different space, which isn't coupled to sensory or motor functions, but rather circuitry as Goldberg Kriech, where you have circuitry talking to itself. So I'm going to project that circuitry over here in pink so you can see that same motif and these two circuits. In green, this is the canonical sensory motor hierarchy, and here is a distributed association network that I'm going to argue is related to higher order thought, then unpack it for you in some detail. Why do we think it is related to higher order cognition? Because of all the kinds of creative studies that our colleagues in cognitive neuroscience have done that have shown how to get these regions to respond. And one really interesting domain that has evolved relates to social cognition. What I'm referring to is social inferences here in the field of social cognitive neuroscience. It's often called mentalizing or theory of mind tasks. These are tasks or functions by which you're able to take the perspective of another. So if you have to have a conversation with a significant other and you want to get through that conversation, you better have a very good sense of what that person's thinking. Humans are extraordinary. Our niche is defined by our ability to collaborate and cooperate. We are food sharers. We are group rearers. Grandmothers, parents work together to rear offspring. And in fact, from an evolution standpoint, this is a critical aspect of our niche. We breed faster than our ape cousins. You might not know that. Humans take longer to develop. When we start hunter-gatherer humans, when we start having children, we have more children faster than apes. And that's because we can, with our partners as a group, rear offspring that are not autonomous while we have another infant. Other species like apes can't do that. Once you're weaned, if you're an ape, you're on your own. Humans are group, we're collaborative. A lot of our brain seems to be devoted to social functions and the ability to represent other people's actions and desires, because that's probably something that's been pushed on in our niche. The way this is tapped into in experiments is you give people, for example, a task where you actually ask them to imagine somebody else's belief. So John told Emily that he's a Porsche. This is an actual stimulus from these early experiments. Actually, his car is a Ford. Emily doesn't know anything about cars, though, so she believed John. When Emily sees John's car, she thinks it's a Porsche or a Ford. I take this control question, which I love, because in all senses and all aspects of logic, it's the same. A photograph was taken of an apple hanging on a tree branch. The film took an hour to develop. In the meantime, a window blew the apple to the ground. The developed photograph shows the apple on the ground to the branch. Same logic, same difficulty, same number of words, roughly matched in terms of how long it takes you to do them. This forces you to go into people, social mode, make social inferences based on your expectations of what people do. This is about inanimate objects in the world. And when you look at a contrast between those two things, you see a distributed network all across the brain that overlaps in some of the idiosyncratic details with these zones of association cortex. This is not one study. This is a meta-analysis of 79 independent studies that have looked at this kind of contrast. So this is a very robust phenomenon. Another way you can ask people to think and sort of detach themselves from the social world, external world, and make inferences is simply if I ask you, and I always say this, if you're drifting off and not paying attention to the talk, I'm okay with that, because you're actually probably using this network. If I ask you to think about what you're going to do later or what you did yesterday, if you're detached from the sensory environment, you probably use snippets and bits of information from your medial temporal lobe memory systems and other systems you have in your brain to build what we experience more or less is a simulation of that other place, another time. We do this all the time. One might argue that humans do this most of the time. If you ask somebody to do a test like that, to remember something from the days before, or weeks before, or months before, you again activate a distributed set of these association networks that seem to fall in these zones that are far from these sensory motor hierarchies. So for all of these reasons, we think that there is support for the ideas that these distributed association networks are used for these kinds of faculties where you detach from the sensory environment and effectively think or cogitate to yourself, whether that's being in the service of thinking about somebody else's thinking or what you're going to do later. All of these require you to detach from the sensory environment and build a mental simulation. This is one of the great feats of primates and human evolution, is to allow us to become mental explorers rather than simply trial and error learners. And we do this all of the time. This led to a functional hypothesis in our end that you could think of these networks as involved in external attention, and all of these other networks are involved in all the ways you can detach from the sensory environment. For example, and for those of you from St. Louis, this is a piece of Forest Park that I used to bike through. You can see here that there was a party and this other individual, and this individual could think back and remember the party. They could think about how they're going to have to clean up and plan. Our guess is that these systems are really about future-oriented cognition, such as planning, thinking about the future, even though we study them most often and remembering. You might think about the perspective of another individual, such as that this woman wasn't at the party, but now you're going to try to coerce her into helping you clean up. You better have a good perspective on this. This is what I mean by social inference, but even more basic aspects of this sort of detachment from the here and now. Even to know where the garbage is when it's not in your immediate sensory environment, you have to have a model to know how you're going to turn and move your body. If you're looking at me and you're thinking about how you're going to move to the door, even if you can't see it, you have a model from your experiences, the spatial layout of this room, of how to build a model of how you're going to move. And so we think that these networks broadly are for that kind of higher level cognition, and I'll get into that in a little bit more detail. We further believe that they're not one network, that there are multiple functionally specialized distributed associations right next to one another in these zones that we missed in our early studies and is missed to date by anatomical studies. And the reason for that is captured nicely in this slide here. This is a former student who did work in imaging here at Harvard. And this is actually the average face from the movie Black Swan. So it's not one person, but it's kind of Natalie Portman-esque. If you look at it, Natalie Portman, when she's an undergraduate here, did work on the brain, but published under her real name, not her screen name. And I actually think all the studies I've been telling you about, largely what we've been doing as a field, is equivalent to this. We blur across individuals, and when you look at this image, you actually learn a lot about the structure of a face. If you didn't know anything about the organization of face, you would learn that there are two eyes, the nose, the relative positions. You might even learn that the difference between men and women, because this is, we could tell this is on average a female. But you also lose a tremendous amount of detail and idiosyncrasies and all the things that make one individual distinct from another. I think human neuroimaging over the last 25 years has largely, with exceptions, I think many of those exceptions come in the clinical translational arena, has largely been about the group average and been blurring and missing details. And so recently we've stopped doing this and shifted to looking at the individual anatomy in detail and actually learned quite a bit more in doing so. So here is that distributed association network that I showed you. Here's the lateral surface of the brain. It actually has components in the midline that I didn't show you until now. One interesting feature, which I'm not going to have time to talk about, is many of them are cortical zones that are tightly coupled to the medial temporal lobe memory system. We've always thought that was a very important constraint, while these networks, such as this canonical sensory motor hierarchy, receives information through the retina. These higher order association systems seem to see privileged or biased information from memory systems, as you might use if you're building models by yourself, detached from the sensory environment. But this is the network you get when you scan 1,000 individuals, project their data as best you can on the surface. It's a very robust network, and you will see the same network if you scan another 1,000 subjects, or for that matter, 50 subjects. So we shifted and we started doing experiments in one individual, a single individual at a time. And so I'm going to put the outline of the network you see from the group, and now I'm going to show you data from one individual that we scanned 24 times on separate days to get our best exquisite estimates of that individual's organization and show you how it compares to the group. And what I'm going to show you specifically is two regions near to each other in this zone in frontal cortex that is part of this group-defined sort of canonical distributed association network you see here. And when you look at one of the seeds over here, you see a distributed network that hits all of these nodes, including this key medial temporal lobe. This is parahippocampal cortex. If you just looked at this result, you might think that it is the same as the group map, but a little bit more precise, as you'd expect when you preserve the details. It's sharper. It's a sharper image of that network. But when you look at a region near to it, you see something quite extraordinary. Across all of the regions of the brain, I'm going to go back and forth so you can see. Pick any region and look at it. Sorry, over here. Watch what happens when you go back and forth. You can see that the region shifts. If you go here to temporal cortex, see this hole right here? Watch it as we go back and forth. They're almost interdigitated. If I summarize that and show you them on the brain, you get a very interesting phenomenon. All across the brain are distributed networks that seem to be at this level of anatomic interrogation distinct from one another and right next to one another. It's as if this entire set of zones is split into two separate things. We've now done this dozens of times and can reproduce this over and over, including this fine interdigitation that, for lack of a better word, I'm just calling this network A and network B, because as of this point, we don't know what they do or what this might mean. But all across the brain, in fine zones, we tend to see these juxtapositions. And I think that's an interesting clue to how they might develop and evolve. But is this an important functional division, or is it something that is just an artifact of the kind of indirect neuroimaging methods? Well, one way you could probe that is with imaging. You can go back and ask, does it externally predict behavior in other contexts, such as when people are doing different tasks? And so if we scan an individual rather than one time, but across many days, hitting the systems over and over with tasks to the point where we can get very precise estimates of individuals, and you target social inferences, what I'm calling theory of mind here, because that's the kind of tasks we did, versus other kinds of tasks where you ask people to remember or imagine the future, these kind of memory-based tasks. What you can find is that these networks are completely functionally dissociable all throughout the brain. And so in black here are networks that we defined, as I just showed you, with functional connectivity. We simply define the networks all across the brain. And then independently, we have people do tasks where they're thinking about the thoughts of other people, social tasks, versus task where they're remembering, and then I plotted that in red and yellow, and it's quite extraordinary. All of these distributed regions that form a network over here overlap, roughly these yellow areas. By contrast, the other network, the second network, overlaps all these regions in red. Remember, these are independently acquired data. It suggests that these large-scale distributed networks of association cortex, there are networks right next to each other, and that these likely reflect functional specializations that probably emerged in evolution and merged during development, by which systems and structures become tuned for helping you deal with things like social inferences or things like imagining what you're going to do later. I don't want to do phrenology here. I don't know what these faculties are and how these systems get specialized, but we can tap them in distinct directions and show they're functionally dissociable by these broad classes of tasks. One kind of theory that is talked about is that maybe the statistical processes of social interactions that you learn about in childhood and that you evolve actually have very different weightings and predictions, and certain aspects of the circuitry become tuned and specialized for them as contrast to other aspects, much like the visual system fractionates and parts of it become more specialized for the central foveal vision and other aspects for peripheral vision. That is something that sculpts after birth by activity, by interactions with the outside world. Higher order association systems may be doing similar things. I think that's a very interesting question going forward. But we can ask very directly, what is the anatomical basis of these networks? And I anticipated it when I talked about the work in macaque from Pat Golden-Rakish, but we can look at it in more exquisite detail. And you could do so looking at the common marmoset. For those of you who have been following this work recently, the marmoset is now becoming a heavily utilized animal system. There's a number of reasons for that. It's a very small primate. Just to put it in perspective, the brain is 180th the size of a human. You can see it here in relation to a hand. This is a marmoset skull. They're very small. They're a regressed species. They're a New World monkey. Most New World monkeys are larger. Many New World monkeys are larger, such as the Cebus monkey, the kind of monkey that you'll see on a pirate's shoulder that's much like a macaque. This is a regressed species. One of the reasons why it has long been utilized, actually we're talking about Clinton, Tom Woolsey this morning. Clinton Woolsey, his father, actually in the 50s or 40s, suggested that people should turn to using these kind of New World monkeys for the reason that the surface of their brain was lissencephalic. It just made it much more accessible to anatomy and physiology. As you can see, while the brain is so small, you immediately recognize the different zones. It is a primate with the temporal lobe, prefrontal cortex, occipital cortex. The basic organization is that of a primate and intact. And so we turn to the marmoset to unravel this anatomy because of these properties, but also because there are databases of anatomic connectivity across the brain in reference coordinate systems that you can do analyses on. For those of you who have been following recently the uptick in work in marmosets, it's for a different reason, but they will converge. It's because they're a genetically accessible primate in the sense that they're very efficient breeders. They tend to have offspring after a few years and will have 2 or 3 babies, so 2 or 3 offspring will survive at a time. So they're incredibly efficient breeders, which makes them accessible to genetic and transgenics. And so that's the reason there's been a lot of research. It's also the case that they have a lot of interesting social behaviors. They're very chatty and they co-parent. That is an example of convergent evolution. Humans co-parent and have offspring very rapidly. This is a New World primate that solved the same niche. It has offspring very rapidly and requires communal, so you have to house them in family units. They co-parent, and I don't think this is a common evolution. This is filling that fast, efficient breeding niche, which humans do relative to our ape cousins. So if you turn to the anatomy of the marmoset, again, just as a touchstone, I'm going to show you a canonical sensory motor hierarchy. This is what I like to do, is to reset before I go into the association zones. Here is an injection to one of the estimated homologs, the frontal eye field. There's actually some debate in the marmoset whether this is the only one or this is the right one, but here's an injection to the frontal eye field, and it's a retrograde label. You can see the projections right here. This is MT in the marmoset, and here is their LIP. This is the canonical, again, sensory motor hierarchy shown in its pieces with one injection across the brain. The other thing to notice is what I also showed you from Brodmann's earlier work, just how much of the brain in a marmoset is visual. This is V1, V2, V3. That's a good portion of the brain. Here is the frontal eye fields. They're quite far back in the frontal cortex of the human. Marmosets have frontal polar cortex, area 10, all of the broad regions we have are prefrontal cortex, but it makes a very small contribution to the marmoset brain. It's become vastly disproportionately expanded in the ape and the human lineage. But this is the canonical sensory motor hierarchy that we think helps you take in information from if I put an injection here, you'd see the retinotopically specific projections to early visual areas by which you take in information from the outside world, project it indirectly through parietal association transformation zones to frontal cortex. Now, what happens when you compare this to the human? Here in green is that sensory motor system. Here is the homologue. This is the MT plus complex in the human. Here it is. And you can see it's just a small portion of the human brain, and here how far back it is in frontal cortex and how much is in front of it, rostral to it, the human, compared to the marmoset. But what's interesting is when you move forward rostrally, anteriorly, into the frontal lobe and move the injection. As you move the injection to dorsolateral prefrontal cortex into these regions that would be blue here, you can see that the projections move outside of MT, anterior here, and also shift in the parietal lobe. So I want to go back and forth between that, just so you can see. Look at what happens here. See how that, you see that shift from what would be the green in the human map to the blue, and that shift into temporal association cortex in the marmoset? This is probably what we often call the cognitive control system in the human now in the anatomy of a marmoset. When you go all the way to the rostral apex of prefrontal cortex, the most rostral zone that falls here in the human, you can see that the projections now completely spare everything in the sensory motor hierarchy and go all the way rostrally over here into the rostral portions of temporal association cortex. I think this is the anatomical origin, the common homologous circuit that existed at least 50 million years now that is expanded and becomes those circuits of higher order cognition that I just showed you. One question is, you can't see it here, but there's a couple of dots here of neurons that are projecting in parietal association cortex. One thing I think that's gotten mixed up in the monkey literature, I think this is a place where Meslam actually makes a mistake. He talks about sensory motor hierarchies, and he groups projection patterns that I think are really part of this transmodal association zone here in the human, these red zones. He groups them with these sensory motor green networks, and you can see why that's easy to do. They're right next to each other, and in fact, they're not that obviously prominent here in the marmoset. I'll show you in a minute that they are prominent, can be identified, but you can see it's really easy if you're going at this from a perspective that this is a visual region. In the marmoset, you really have to want to believe there's something else there to even notice it. I'm going to unpack this in detail because I think it is critical to be specific here. This is human association cortex, this network in 25 individuals. I've now flattened, unraveled it, flipped it so that's more like you'll see in the monkey anatomy literature. This actually preserves the relationships. Here's the distributed network in the lateral surface. Here it is in the medial surface. And I want to show you where all of these regions are in the marmoset brain. To do that, I'm going to actually have to show you a completely different representation. This is a flattened representation of the marmoset brain. Here is occipital cortex V1 back here. Here is the temporal pole over here, the front of the temporal pole. Here's parahippocampal gyrus, medial temporal lobe memory system. Here is the frontal lobe, the apex of cortex. And so just to orient you, here's auditory cortex, here's motor cortex. I don't expect you to keep track of these things, so what I'm going to do is place blue diagrams around the 5 zones in the human that are prominently in this distributed association network and show you where they are in the marmoset. I'm then going to show you a single injection in the marmoset in frontal polar cortex. And you can see that it receives projections from these distributed zones, and critically, those distributed zones selectively, not from MT. If you move to FEF, you get dramatic projections from MT. Here is an average of 8 injections in the marmoset. The marmoset has a distributed network of these sort of transmodal zones that have projections to these rostral apexes of prefrontal cortex that is anatomically distinct and interdigitated with their sensory motor hierarchies, just like the human. What I think happened in the primate lineage is somewhere on becoming a primate, there was a new axis that emerged, probably, well, I should say, speculatively, from the rostral apex of prefrontal cortex, but somewhere in these association zones. And you can see sort of the evolutionary divide and the separation of that circuitry. This might have been the opportunity for evolution to start sculpting and differentiating circuits that are no longer bound or tied to the sensory motor environment. Goldman Rakesh, her brilliance, noted this early on. She noted that these distributed association networks didn't fall in sensory motor hierarchies and they looked different. And she actually wrote at the time, if this is so, then traditional ideas of hierarchical processing that may apply within some systems is not the dominant functional organization of the association cortex. This is interesting because it's hard to convince people of this alternative organization, its implication. I think it makes up most of the brain, but it's hard to study and really hard to study in macaques. And so there's a bias to sensory motor experiments and thinking, but actually, from the anatomy, you can see these. And she went on, rather, higher cortical functions seem to be carried out by a finite number of dedicated networks of reciprocally interconnected areas. And I think that this was prominent at least 50 million years ago because of our evidence from the marmoset, and that's our common ancestor occurred at about that time. So I don't want to suggest that humans are the same as marmosets, and I think that interesting to think of what happened on the way to becoming human. And I think that the main thing, this is from Marcelo Rosa's laboratory, an estimate of cortical expansion from the marmoset, the macaque, and the human. The first and obvious thing is that our brains are much larger and our association zones are disproportionately larger. We've written about embryonic constraints that might dictate this. I don't think you have to have anything special happen to get disproportionately larger association cortex. I think if the sensory motor regions are highly constrained by embryonic axes in development and you expand up the brain, you might simply get larger association zones. And what we're seeing when we look in the human are specializations of these distributed circuits that are well present in monkeys, but maybe not differentiated, and surely not differentiated to the same degree for these faculties, such as remembering and social inferences. But the most interesting aspect that relevant to Geschwind is I've left out, and it's often not thought of this way, there's actually a third component to this distributed association network, and it is what we commonly think of as the language system. I know we think of the language system by its nodes, seeing what happens when there are lesions to this broad region in posterior cortex or in frontal cortex. But here, when you look at the functional organization in terms of topography, it actually looks like just another specialization of this distributed association motif right next to it. And when I say specialization, I want to be really concrete here. That what we're thinking of as sort of Geschwind's language network might be another component of that. You could probe that, see its specialization. So if you take an individual, you place them in a scanner, you do a task that Ev Fedorenko, who's now at MIT, where you show people sentences such as a rusty lock was found in I can't read it, the drawer. But who helped me out there? So can you help me out here? So you have people do this, these sensical sentences versus these nonsensical sentences. You ask what parts of the brain. You will produce a map shown over here. This is a map in an individual of them doing that language contrast. It's distributed nodes. Over here on the left is our estimate of networks in individuals purely from their functional connectivity. And I'm highlighting in yellow what we think is the language network right next to these other networks. And the outline of that network is shown here in black. And the independent language data is shown over here. It's extraordinary how well they overlap, even across idiosyncratic parts of the brain. If I showed you the midline, there's many zones of the language network that we don't think about, probably because they're not prominent neurological syndromes. But nonetheless, here is that distributed network. And here you can now look at its anatomic selectivity by asking, just taking these networks you define in an individual before you have them do any task, and then you ask, what is activated when you do a language task. And you can see here the prominent activation during language, and the networks right next to it on either side do not respond. Here it is in another person, here it is in another person. We've done this now dozens of times. These distributed networks that are right next to each other in this chronic motif, by the time you become an adult, are extraordinarily specialized. And this is probably what happened on the way to becoming a human. So just to end with a little bit of speculation, here we have this language network. It's fascinating that it's right next to networks that you use when you think about whatever other people are thinking. If you think about this cooperative niche, we are good at imagining other people's perspectives, but we're good at communicating and talking. Probably the human niche is about that collaboration, about cooperation, these interactions, and you see these higher order association circuits fragmenting and specializing for these faculties. What we think is that it's likely that these networks are defined, somewhat, rigidly specified by gradients and properties to develop an embryo that a proto-organization at some level forms, but then protracted probably from, the first several years of life, you see the specialization that we now know of as the adult form in these regions. This is hard to actually glean from lesion studies, because when you damage some of these regions and you see the language impairments, one does not ask about subtle social influences, and maybe they're not as specialized and not as clearly defined. But from our perspective, we think all of these networks are actually juxtaposed right next to each other, forming a general motif. So, what might have happened in evolution as there's expansion is all of these zones, which were well represented in our primate ancestor 50 million years ago, became vastly expanded and seem to have taken on this extraordinary specialization for higher order cognition. I just find that absolutely fascinating. So I wanted to end with this, because when I said that our work has been influenced by and is led from, we very directly lean on the ideas that Gershwin set in motion. And in fact, it's interesting to see it was come full circle. The slides that I showed you there with Gershwin's I did not put them together for today. They're just actually part of our story. That's how it has been evolving as we've now unraveled with new tools, refinements of them, and convergence with other tools as we've unraveled large-scale network organizations. So thank you for your time. And I wanted to thank some of my colleagues who have done this work. Rod Braga, who did all the beautiful, exquisite work. We pushed the limits of within subject functional localization. Lauren DiCola, talented graduate student, who's now been using tasks to probe the functional specialization of these circuits. And Daniel Margulies has been my partner in crime on all the marmoset work, which draws heavily from the cases that Marcelo Rosa put online. We didn't do the injections ourselves. We've just been mercilessly pulling all of the data from online open repositories and analyzing it. So thank you very much.
We have time for a number of questions, and then there's going to be a reception afterward in the CLS7 conference room. Everybody's invited to have something to eat and to talk further with Dr. Buckner. Before we go to Mo, I just have a quick question. So your drawing of the Gershwin language circuit reminded me of his work where he compared it to a praxis circuit that was essentially a parallel circuit to what he thought was the language circuit. Have you looked at praxis? Is the circuitry for that also similar to this? Is it all in those domains?
So a great question. When you look in the marmoset, I unfortunately don't have the slides as backups for this. When you look in the marmoset, and you start to walk from primary motor cortex to their caudal premotor area, to their rostral premotor area, to prefrontal cortex, frontal eye field, and then to the rostral, what you see is just a hierarchy of networks that have all the same form. And if you look on their brain, it just goes from anterior regions to the posterior regions, wider and wider and more distributed. So your impression is there's just a hierarchy of networks with the same motif. So when you ask, are they next to each other? They're all neighbors, with their sort of caudal rostral gradients. The functional point that we're trying to make, though, is that can be deceptive, that at some point, these higher order circuits broke off and became parallel. And Mike Fox has actually been one of the ones to show, key point in this. It's not just that they're another network. There might even be competition between these sensory motor networks and these higher order networks. In evolution, they might have broken off, through just the developmental gradient, because there's maybe no other way to develop these things or evolve these things. But at some point, they become so functionally specialized, they might even be competitively interacting.
But if you chose the task properly, you might find neurons participating in more of them.
You know, you often find things that are more than one network. In fact, when I showed these things, you might get the impression that if you do a task contrast, it activates a network. That is not the case. If you do a task arbitrarily where you show visual things and people hear things and they're moving, you'll see multiple networks all over the brain at the same time. The task contrast I'm showing have been the ones that have been winnowed down to isolate a network. That said, it's hard to find a task that simultaneously activates some of these higher order association networks at the same time as simple sensory motor networks. I know in a dynamic world we're using both of them, but it could be we're using them dynamically over time, and that at some deep level, they may be different modes, almost like we have muscles. I mean, and I'm sure there'll be an exception. We have antagonistic muscles that could be thought of as working against each other. You can set up a situation where you flex them both, but that would capture, the sort of dominant aspect of their function where they're specialized and working antagonistically. These networks, some of them, do work like they're working antagonistically across broad ranges of situations. And that's an interesting feature. In dynamic movement, you'll see all of them used, but in isolation, they might be better thought of as specialized and working against each other.
First of all, great talk. Thank you very much. I'm going to bounce off this question a little bit, which is how much of this functional specialization or the segmentation of networks that you see is a product of the tool that you're using, where you're looking at correlated changes in workflow, and by definition, it can only be correlated. It can't be correlated with multiple different things simultaneously easily. But if you actually did electrophysiology, instead of distinct networks, you might see 60% of the neurons in one area responding to task A, 40% task B. At the fMRI level, it looks like these are distinct areas, but they're actually completely interdigitated. Mm-hmm. And I think there is some evidence for that from the macaque literature in working memory, for example, where if you look at different functions and rule generation, for example, those neurons are actually interdigitated. It just may be that 60% in one area are working memory, and the other 40% are rule generation to you because of the nature of the fMRI study that you're doing. These look discrete, but in reality, they're interdigitated and dynamic.
Great question. And that stems from actually, I present the anatomy of Goldberg Keisha, and one of the great debates that emerged in these regions, and her idea was specifically, I didn't go into this, is that these frontal zones were content specific and gained those properties from their posterior zones that she got. And then Earl Miller, a number of clever experiments, John Assad, others, I mean, just absolutely stunning a work that challenged that showed if you recorded from these regions during the rule phase and then later they become domain and content. And so that kind of ilk is now suggesting, well, maybe these things are not as special as you're saying. They're all multi-determinant, and the specialization comes from the technique. I think it's an interesting idea, and I'll give you two answers to that. I think I would not conflate the fact that there might be specialization, interesting features in local territories of cortex with the idea that might be broad specializations across these large regions. I think these large regions of specializations do exist physically because of how well they predicted the anatomic connectivity in the macaque and physiology in human recordings at the broad level. So I don't think that's going to be right at a broad level. I think this is probably carving nature at its joints, and it is not technology dependent. So I want to make that point extremely clear. Now, to speak to the origin of this very interesting debate, which I followed, so I was weaned on that as a graduate student and as a postdoc. It's interesting. I think the debate might be reframed. I think what Pat was doing is overestimating the sensory content aspects of these transmodal association networks. I think she was already in these association networks. She was in these higher order arenas, which is interesting. In any moment, they'll help you deal with anything. And so I think she was overestimating their sensory capacities, and really, they're much more like those abstract properties. But in fact, they still probably have a processing domain that is distinct from this region an inch over. And so I think that's an interesting discussion about what those regions are doing in sort of the evolving context of that debate. But I would not use that data to undermine what I think is going to be a method an experiment, independent observation, but the brain has large, broad networks. And I say that because I can march you from caudal rostral and the marmoset with retrograde labeling techniques and with functional connectivity, and you see the same thing.
I've always been interested in tuning. It's the idea how in training actually then tunes the sensory system and nervous system of the system. When you teach people about what the kidney looks like, then when they look, they're looking for something or you're feeling for something. So it's really the cognitive behavioral tuning the other. Is there any kind of research about that or how that works?
Yeah, another great question. So in real world and complex tasks and in dynamic things, we often study these things statically, but in the world, we evolve. We look out to the world, we think, we kind of go back and forth very quickly. There are surely interactions. And so in some sense, this talk was more about splitting to make the point these separate networks, but then you get the question, well, how do they interact? The place where there's been a lot of work on this is actually in one of the domains that I referred to here in remembering. So I showed you how these networks take in inputs from the medial temporal lobe memory systems, but if you close your eyes and remember what somebody looks like, what something sounded like, we know that there are interactions with sensory systems, and you do see those. If you close your eyes and remember something you saw yesterday, you will see these regions come online with some of these extra stride regions. So that is a boundary condition where you do see a kind of interaction. But broadly, they tend to be competitive. But there are clearly places where, and how they interact, I don't know. But in learning, surely they are. They're going back and forth, and that expertise, at least in humans for now, deep learning algorithms and computers soon.
You could study people before and after training.
We do, often.
Very interesting to see how they change, how one individual can.
Yep. Yeah, that's something. Actually, we have not done that in the individualized arena, but that would be very interesting.
So thinking about kind of clinical applications, when you have the scans of the individual subject and you did 24 scans, how many did you need before you started to converge? And then what do you think caused that differences from scan to scan?
Well, so things. When you look at one group of scans versus another at that level, they look very similar. There's just signal noise. The properties are such that as you have less and less data, they're noisier and noisier estimates. When you ask, well, how many scans do you need at a very practical level? I always answer that by saying, how many scans do you need for what? So after 8 minutes, I can now, knowing the details of this, see some features of it. In parietal cortex, I usually can pick up the separation of network A and B in parietal cortex. At 24 scans, we're still debating a couple of details on the midline. So finer and finer juxtaposed details, and I'm certain with our crude resolution, we're missing lots of stuff. So both resolution in terms of more data are still helping us discover more, but when you have 6 minutes of data, like you have in the UK Biobank, we've interrogated that data, there's 20,000 subjects, very little data. Now knowing what we know about the sharp details in individuals, using that as priors, we can see aspects of that in the larger group data. So it depends on for what.
Wonderful talk. A quick question. You mentioned how the visual system is sort of wired prenatally, but then is sort of refined with visual experience in early childhood. Then you talked a lot about sort of theory of mind networks that are also there before birth, but are presumably also wired with learning in early childhood. I imagine the domain of dysfunction there would be psychiatric disorders where early childhood disturbances prevent that formation. People started to look there, and do you think there's any traction that can be gained by using neuroscience to look into?
Yeah, so a great point. So I think, again, I tend to have a perspective that evolution works simply. Like there are probably some really interesting innovations that occur every once in a rare while in evolution. But by and large, the rules of wiring, the competitive dynamics that form wiring probably are largely conserved, from rodent to human. So when we look at the visual system, you can see the competitive dynamics, early visual cortex, juvenile and weasel, but now Marge Livingston beautifully shows how these higher order extrastriate regions like the face selective patches start to form during the first year as there's visual experience. The scaffolding is put down by the retinal waves and other features that occur before birth, but that scaffolding is then further sculpted by dynamic interactions. And you see that unfold in the visual system beautifully now. Our guess is that the more neonatinous association cortices would have a more protracted development. And maybe that's the evolution. As the brain got bigger, more of cortex was neonatinous, but it still is going to have the same competitive, activity-dependent kind of formation of specialization. And the guess is that does form now not early development, but over the first 3-5 years, when you start to see emergence of social faculties and language. And our guess is that when you have breakdowns in that, you do see the early neuropsychiatric, language delays, autism. I don't think it's a coincidence that language, social, and these other faculties are all potentially tied together in some of the developmental disorders. One could imagine the disruptions in the proper specialization of these networks for a variety of reasons, whether that be a molecular break, a rate-limiting, transcription factor like you see in Rett syndrome, or just other reasons that cause you not to interact with the outside world. To the degree the exquisite specialization doesn't form, you're going to have difficulty with higher level cognition, and social function and language are very fragile, very high level faculties.
So I think we've run out of time here, but let's, first of all, thank Dr. Ruffner, and secondly, move across the street to continue this.
Thank you.